Axel Borst. Some of you, many of you were here on Thursday, so I don't need to repeat the fact that he's the lord of the flies, as you heard from Tali, or king of the flies, as Tali called it. Just wanted to add two comments on this king of the flies. As you heard last time when we spoke, that there is this kind of a broad view with many techniques, from modeling to molecular biology, from anatomy to behavior, single cell, anything you want. So this is, I think, an example for all of you that you can think of yourself as inside yourself, multidisciplinary. I think Axel is really an example of an experimentalist who is not afraid of writing a paper in Nature Neuroscience on information theory. He can talk with Chaim today on compressed sensing. He can talk with me on cable theory. Not only talk, understand. So this is really a fantastic example of a modern neuroscientist, in my mind. Really an example. Not to speak about the 160 papers that he wrote, many in Nature and Science, but this you can look at the internet. So that's something that you should be aware of, an example of what I think a modern neuroscientist should look like. Not only in the way he looks, red and all that, I mean, look like scientifically. The other thing I wanted to mention with respect to Axel, there was a point in his life that I was very disappointed because he was grabbed by the Americans, by Berkeley, and I was thinking, why did the European, German, left him out? How can somebody give him up, so to speak, and allow this far place to take him? But eventually, of course, they decided correctly to make Axel a Max Planck Institute director in Munich in the last 12 years. Is it 12 years now?
It's 11 years.
11 years, highly successful. So I'm very glad because in some sense this means that he's very close to us. He can come easily, we can go easily. And this is exactly what I wanted to tell you as the final line. We now have together three of us here in Israel from ELSC, Chaim, Martinsried, and myself, and Axel Borst, Tobias Bonhoeffer, and Bert Sackmann were nominated, so to speak, together to erect a new Max Planck Center, which is a new phenomenon worldwide. It's not an institute, it's a center. The center here is part of ELSC, the center there is part of Max Planck, so we are starting to collaborate. I'm sure you will hear a lot about this in the future. So thank you very much, Axel, for coming. The stage is yours.
All right. So thanks again, and thanks again for the very kind introduction. Well, in my department of systems and computation and neurobiology in Martinsried, Munich, we study information processing in single neurons and small neural circuits. And as a tractable system for that, we picked the fly visual course control. Now, fly visual course control is summarized in this slide. When the fly is buzzing around, it creates by its own movements the images of the environment to flow across the retina. And we can quantify this image flow as an optic flow, which is a vector field indicating the direction and the velocity of each image pixel at each point in time. So this optic flow is perceived then by the retina of the fly and then processed by several layers of neuropil. And these several layers have names, and I'm going to use them a bit more often today than 2 days ago. The first layer of neuropil, where the first synapses are made of the photoreceptors, is called the lamina. Then it is followed by a more complex neuropil called the medulla, the lobula, and the lobular plate. As the single most important computation between the photoreceptors and the lobular plate, local motion detectors calculate these local image vectors of the optic flow. So at the output of the medulla, this optic flow is represented by the activity of local motion-sensitive elements, and we call this representation neural flow because it is not really identical to the optic flow, right? Now, at the level of the lobular plate, these activities of these local motion-sensitive elements are pooled and integrated by a set of, in case of Drosophila, 20 to 25 different large tangential cells. Now, each of these tangential cells has a unique receptive field that matches a certain optic flow, and I have given some examples 2 days ago, and today I just want to show you some measurements that Max Josch in the lab had done on an HS cell. So that is the receptive field of an HS cell, again, sort of represented in spherical coordinates as a vector field, and that would be like the receptive field of the VS6 cell, and you see this curled structure indicating that it will respond maximally when the animal is rotating, right? So the activity of these lobular plate tangential cells is then used by the fly to control the position of the head and the steering maneuvers of the wing. And I have shown to you optogenetic evidence that this is indeed the case. So when we optogenetically stimulate, these cells, the 3HS cells, then the fly turns the head towards the stimulated side, and it also exerts a torque in the direction of the stimulated side. So we know that activating these cells is sufficient to elicit optomotor responses of the whole animal. Now, today I want to focus on a single computation, and that is elementary motion detection that is going on between the photoreceptors and the lobular plate. Now, why is that an interesting computation? Well, if you take a white bar and move it to the right and to the left in front of the fly's eye, and you take an electrode and you stick it into the photoreceptor axon, then you will see that the photoreceptor signal is the same no matter whether it moves to the right or to the left, right? A single photoreceptor, as I said, cannot discriminate between rightward and leftward motion. Now you take this electrode and you move it a few synapses downstream into the lobular plate cells. You do the same. All of a sudden, you get direction selectivity. In this case, the cell responds with a depolarization for rightward motion and a hyperpolarization for leftward motion. So how can that be? You go from non-directional signals of the photoreceptors to direction selective signals at the level of the lobular plate. In between, some computation has to take place. This computation has been formally described by a model called, after its inventor, Werner Reichardt, the so-called Reichardt Detector. That is an algorithmic model that describes how you go from non-directional signals at the level of the photoreceptors to direction selective signal as we find them at the level of the lobular plate. The Reichardt Detector is built from two mirror-symmetrical subunits that share the same input, and within each subunit, the signal from one photoreceptor is temporarily low-pass filtered and subsequently multiplied with the instantaneous signal from the neighboring photoreceptor. That's repeated in a mirror-symmetrical way here in this right subunit. Then you take the output of both multipliers, you subtract them, you end up with the perfectly direction selective signal. This Reichardt detector has been extremely powerful in predicting the responses of both the optomotor response at the behavioral level as well as the neural responses at the level of the tangential cells. As innocent as it is looking, it produces a number of sort of non-intuitive predictions that we all verified in experiments. Most notably, and so to say, the latest example for an interesting property that comes out of this detector is velocity gain adaptation. I'm saying that because Nama Brenner, when she was at that time with Bill Bialek and Rob de Ruyter in the United States, discovered that the tangential cells shift or change their velocity gain according to the range of velocities that you present to the animal. When you have a very small velocity range, so only small velocities occur in a sort of a statistical sense, so you wiggle back and forth, then the neuron is very sensitive. You can calculate a very steep gain, steep slope in the response velocity function of H1. That is one of the tangential cells. When you cover with your statistical stimulus a broad range of velocities, the gain is very low. That means now H1 can cover a large range of velocities and still sort of transmit this information to further down postsynaptic signals. That was published in the year 2000 as an experimental finding. And when Chaim came to Munich, I think this was like 8 years ago, we discussed that, and then we worked on this Reichardt detector, and we found, to our really amazement, that it does not require any adaptation of parameters here. This model produces this velocity gain adaptation automatically. That's just to tell you that it's so important. Even when you think you can intuitively grasp such a simple-looking model, it's very important to do the actual modeling or calculation. Right? So in order to just qualitatively convince you that this really is a good description of what's going on in the fly visual system between the photoreceptors and the tangential cells, I'm going to show you one example where we used such a statistical velocity profile. We used a grating pattern that was moved back and forth, with this velocity profile. And at the same time, Hubert Eichner, a former student in the lab, recorded with the tungsten electrode the action potential activity of the H1 neuron. And you can see that whenever the neuron, whenever the pattern moves in the preferred direction of the cell, then you get a large increase in firing rate. And when the pattern moves in the null direction of the cell, then H1 ceases to fire and fires no action potential. And then Hubert modeled H1 by an array of Reichardt detectors that are spatially summed and feed into a leaky integrated fire neuron simply to get action potentials out. And this is the result of the simulation. And if you compare the actual data with the simulation, you will agree with me that this is a very good model, right?
Why are there different detectors on the... Is it necessary? There are several detectors.
Yes. Okay. So what you need, a Reichardt detector is a local motion sensor, and what you need is an array of motion sensors covering the whole visual scene. So that sort of is one spatial coordinate here. That's the x-axis. We didn't do a two-dimensional because in the y direction, there is nothing sort of, it's simply then the same as here. So we did here a one-dimensional array of Reichardt detectors feeding into this leaky integrated fire neuron. For the specialists amongst you, if you look at the output of a local, of just one Reichardt detector actually, then this is different from the array because you see a modulation at the output that does not only depend on the actual velocity of the stimulus, but also on local luminance values. Now, integrating these different Reichardt detectors look at different spatial phases of this periodic grating, and then integrating over an array of them then sort of cancels out those modulations that are due to the local luminance modulation, right? So you need an array of Reichardt detectors in order to get a good velocity estimate. So I told you this is a formal model to describe the transformation between the input of the output of the photoreceptors and what's going on at the lobular plate. So, of course, we're interested to see sort of who is presynaptic to these tangential cells.
Do you get the filtering parameters independently?
Yeah. So we knew them. And actually, you get a very good fit if you add here a high-pass filter in addition. And so this is, again, another project that I did with Chaim and one of his students. So let's look at what type of neurons we find presynaptic to the dendrites of the lobular plate tangential cells. So here again you see the optic lobe. Now we take a horizontal section of the optic lobe and we look at the different types of neurons that we find there between the photoreceptors and the lobular plate tangential cells. Well, first of all, you see that the photoreceptors in the retina, the photoreceptor axons of R1 to 6, they all terminate here in the lamina. R7, 8 go straight through the lamina, terminate in the medulla. They're not important in the present context. Here, the signals of photoreceptors R1 to 6 are taken up by lamina neurons, which take the signal here. This is the dendrite, and they feed the signal into the medulla. There are several of the lamina cells, and I will talk about them later. Within the medulla, which is actually a fairly complex neuropil, you find intrinsic neurons that do not leave the medulla. They simply connect different layers of the medulla. You find neurons that take the signals from different layers within the medulla and branch out in a Y-shaped fashion, TMY, and then send the signal to the lobula as well as to the lobular plate. There are other cells that simply go to the lobula, and there are cells like the so-called T4 and T5 cells that take the signal either here from the medulla, bring it to a certain layer in the lobular plate, or do the same thing with the signal here from the lobula, bring it to the lobular plate. I will later talk about these T4 and T5 cells in much detail. Well, interestingly, the first account of all the different cell types that are in the fly optic lobe has been done by Ramon y Cajal, taking the staining method of his competitor, Golgi. And he, published in 1915 this book together with Sanchez, where he, captures a lot of the different cell types that we find in the fly optic lobe. For example, the T5 cells connecting lobular and lobular plate, or the T4 cells connecting the medulla to the lobular plate. And he drew them in astonishing detail. For example, for the specialists again, you see that there are two processes leaving this dendrite here, one leading to the axon terminals, and the other one being the cell body fiber terminating here in the cell body layer, posterior to the lobular plate. So it's really amazing that Ramon y Cajal saw all these details. But it wasn't a complete account. A complete account was then provided later by Fischbach and Dittrich in the late 80s. I just wanted to show you one example of how we stain these cells nowadays using genetic methods with specific driver line in Drosophila. For example, you find in this Drosophila market square, someone who gives you a driver line for TM2 cells. You do immunostaining GFP expression, and you get this beautiful image of just one cell type per column called the TM2 cell. And then you cannot just visualize them, then you can do something with these cells. This photo has been taken by two grad students in the lab, Etienne Saab and Matthias Meyer. Now, as I said, a full account of all the different cell types we find in the fly optic lobe has been provided by Fischbach and Dittrich from Golgi stainings again, and they describe about 100 different cell types, which all occur for each column once, right? And here, all of them are drawn one on top of the other. To appreciate the real complexity, you have to imagine that here they are drawn, so to say, apart, but you just take them and squeeze them, so you pack them all within a single column. Then you are at the real situation in the fly optic lobe, and this is what we're dealing with, right?
How many cells altogether in one column?
About 100.
So one copy from each
Yes.
cell is cell type.
But
So each cell is cell type.
yes.
But then it repeats.
Yes, exactly. That's a very important point. What do I mean with cell type? Right? It's not obvious. So this is the situation that we're dealing with. On the other hand, we have this nice, clean, fairly simple description of this local motion detection by the Reichardt model, right? So the Reichardt detector, as I said, describes the processing in formal terms using, things like, a low-pass filter, a multiplication, a subtraction. Doesn't talk about neurons. So in a sense, it's like a black box, to us. But how these things relate to each other has been an enigma for a long time, because these cells are way too small to record from them directly. So you have to imagine that the response properties of most of these cells is completely unknown and has been unknown for about 50 years now. No one has ever been able to record from them and relate this computational structure to these neural cell types. So finding this relation, the neural implementation of the Reichardt detector, has become like the Holy Grail of fly motion vision to me and a couple of other labs studying in this field. I cannot tell you how embarrassed I was over several decades almost, that each time I was giving a talk, I was talking about those local motion detectors providing input to the tangential cells. At the end of the talk, you for sure get the question, which cells do this computation? Each time you shrug your shoulders, say, I don't know. You say, gee, one day you need to find out about it. And so when I moved to Munich, we started to ask the little fruit fly Drosophila to help us open this black box, right? And my talk today is about how far have we come opening this black box, relating this computational structure to its neural implementation. And we took Drosophila because of the power of genetics that it offers. And I'm sure many of you are aware of the GAL4 UAS system that Brandon Perimon introduced to the field in '93, where you have a driver line that basically determines where in the nervous system something is being expressed, and an expression line that says what is being expressed. And the what is being expressed in most of the cases today will be a blocker of synaptic activity of a neuron, and that is the Shibiri TS gene. Now, Shibiri TS is the temperature-sensitive allele of a gene that codes for dynamin. And dynamin is used in vesicle recycling. It's a GTPase that is used to strangle off this vesicle when it's being recycled. And it is a dominant negative allele, so you can use a wild-type background, and you add the transgene, Shibiri TS, and upon a temperature shift from room temperature to 35 degrees, the activity will die out. That means within 2 minutes, there's nothing, no vesicle coming out, and no transmitter being released from that neuron that expresses the Shibiri TS. And the nice thing is that you can use the same flies with the identical genetic background. In one case, you simply shift them to this non-permissive temperature. In the other case, you leave them at room temperature, and then you do the experiment. One case is your control fly, the other one is the experimental fly. Right?
What about gap junctions?
That is something you cannot block with it. That's the downside. Right? So, and in fact, there are other tools that we also use, like potassium inward rectifying channel that's called KIR, that sort of clamps the cell at its resting potential. And this then is expected also to spread over gap junctions. So, in fact, we get different results depending on whether we use Shibiri or KIR, and we can explain that by the fact that KIR also inactivates neurons that are electrically coupled with it at the same time, whereas if you express Shibiri and this neuron has gap junctions as well as chemical output synapses, only the chemical output synapses are affected. Right? So that's another important point to keep in mind. Now, the other sort of essential point in the analysis that is coming is that Max Jösch, when he was a grad student in the lab, managed to do patch clamp recording from the tangential cells in Drosophila. So this then later allowed us to combine electrophysiology, that is recording from tangential cells, with genetic inactivation and blocking of presynaptic candidate neurons in one experiment. And that shows recording from a VS cell that Max did, where you see that the S cell shows a nice direction selective response. It hyperpolarizes when there's upward motion, and it depolarizes via some action potential in response to downward motion. Now, in order to sort of proceed in a nice way, we first wanted to ask what cells of the lamina provide the input signal to the motion detector. And as you can see from this scheme here, there are 5 different lamina neurons called L1 to L5 that all sit postsynaptic to the photoreceptor axons in the lamina and send their signals in parallel to different layers within the medulla. So that was sort of a very sort of obvious question, which of these cells is providing the input signal to the motion detector? And as I said, the paradigm that we're using is then to record from the tangential cell while inactivating either L1 or L2 or both of them, move something in front of the fly, preferred direction, null direction, and see, do we get a normal motion response or is the motion response impaired when we block L1 or isn't it, right? And of course then you need a driver line, for the specific cells that you're asking. That is the driver line for the L1 cell that shows, ramification in medulla layer 1 and 5, and that is a driver line for L2 cells that shows ramification in medulla layer 2. These images have been taken by Sham, postdoc in the lab. Now, first important data slide. It's a little bit overloaded, but I think you need all these details. What you see here is the response of the tangential cell in millivolt as a function of the grating at various contrasts from 0 to 100% contrast that is moving in the preferred direction or the null direction of the fly. Black is always the control. Now what you see is that, control flies, sort of show this nice depolarization response for preferred direction. They hyperpolarize in response to null direction. And as a function of stimulus contrast, you see that it saturates with increasing contrast. If we block L1 and L2, both of them at the same time, the motion response is gone completely. So you immediately can conclude that L1 and L2 both represent major input to the motion detector, right? Now, if we block L1, then we see that we can reduce the preferred as well as the null direction response over the whole contrast range by about 50%, right? If we block L2, with using 2 different driver lines, again, we can reduce the preferred and the null direction response over the whole contrast range, maybe a little bit less than by blocking L1, but still significant, right? So we know that L1 and L2 represent the major input, but from these data, we had no clue in what respect L1 and L2 might functionally represent different, qualities of the stimulus, or whether it's just redundant input. And we've been going through a whole list of ideas that maybe L1 feeds into the horizontal system and L2 into the vertical system, maybe L1 in the low contrast, L2 in the high contrast range. Nothing of these ideas ever fitted. Until we came up with an idea that we borrowed from the vertebrate retina, and that is maybe the signal is being split into positive contrast and negative contrast increments and decrements. So splitting into an on and an off channel, like what we see in the vertebrate retina. Of course, we would not see that splitting here because we used a grating that has brightness decrements, brightness increments, brightness decrements, brightness increments, right? So how can we test for that? And this was the birthday of our on and off edges that sort of brought a lot of insight and excitement in the lab. So now what we used on edges and off edges and tested again L1 block flies and L2 block flies And here you see the time course of the average response of the control slice for a moving on edge in the preferred direction and a moving for different control slice. And here you see the different control slice responses to a moving off edge. And it started here at 500 milliseconds, right? So now we expect something different to happen when we block L1 than when we block L2. So let's see. When we block L1, the response to the moving on edge is completely gone, while the response to the moving off edge is absolutely wild-type-like, right? If you block L2, the opposite is happening. Now, blocking L2 leaves the on-edge response unaffected and reduces strongly the off-edge response, right? I mean, we published that in a prominent journal, and that was, part of the thesis of Max Josch. The actual data that I'm showing here were collected by Franz Weber a month or 2 months ago, because for some reason we, did some other experiments with L1 and L2 block flies. So I told Franz, please repeat these experiments. If you publish it, make a big fuss around it, you better be sure that it's really true. And so I was extremely satisfied that Franz found the same thing that Max found 2 years ago. Yeah?
Is it for a single direction?
It's actually preferred minus null direction, what I'm showing here.
So a single preferred direction or average overall?
Oh, so, okay, so we had, here you see the average data of HS cells. So for them, preferred direction is front to back, and downward motion for the VS cells. But we also analyzed them separately. There's no difference. So we pooled the data. And the effect is sort of as evident for preferred direction as it is for null direction. And then so we pooled for HS and VS cells, and we pooled, by taking the difference between the preferred and null direction response.
What's the behaviorally this slides when you block L1 or L2 in terms of behavior?
So yeah, I can. So we haven't done that extensively, but I can tell you at the end a little bit about it. Tom Clendenin had very similar findings by looking at the behavior of L1 and L2 block flies, and also found a selective impairment of L1 block flies to on edges and L2 block flies to off edges, right? But I can also talk about where his lab and my lab have a disagreement, right? Now, so as a summary from that study, I have told you that we conclude that L1 and L2 provide the major input to the motion detector, and they are functionally segregated by L1 carrying the on signal and L2 carrying the off signal of that. Now, that has sort of a number of different sort of implications. First, it seems like a general rule or evolutionary bestseller, how I call it here, this splitting of a sensory input signal into an on and an off. As I said, we find it in the vertebrate retina. Frank Werblin and Dowling published that in '69. Last year, a paper came out about Drosophila thermoreceptors from Zucker's lab that shows that here at the base of the arista, you find cold and warm receptors that, one is going up when it's getting cold, the other one is getting up when it's getting warm, and so they really operate in counterphase. And from Corey Barkman's lab, there's also publication from C. elegans that in chemosensation, the same is true. There are neurons in the worm that sort of signal the onset of a stimulus, others signal the offset of a stimulus. So it's something that you find across different species. For me, the most important thing was that splitting the signal into on and off provides a very easy way of multiplication. Imagine you have these two photoreceptors sitting next to each other, and the grating is coming by. So, if they sort of signal both increments and decrements within one line, you will have that picture. Now, then later, you want to multiply them. So what do you need? You need a unit that increases supralinearly when both inputs go positive, but the same unit should also go positive if both inputs, go down. And that is something that sort of caused a lot of headache to me in previous years. Now, if you split the signal into on and off and then multiply in different channels, this problem is alleviated a lot. So you take the positive signals, feed it into one multiplier, and you invert the negative signals, represent them in a different channel as positive signals, and feed them to a different multiplier. Now, all of a sudden, your nonlinearity problem looks like that. You need a simple supralinearity here. And, this sort of the sign rule of multiplication is taken care of by splitting the signal into an on and an off channel. As it is with any kind of discovery, it leads to a number of next questions. And I want to go through 3 questions now. And first is, how is the pathway specificity achieved? So there are recordings available from the lamina cells, L1 and L2 in large flies from Simon Laughlin's lab. They have been published already in the 80s. And both L1 and L2 cells hyperpolarize the same way when you depolarize the photoreceptors, by just shining light onto them. And this is just shown for 2 light levels here. But the signal from L1 and L2 is sort of indistinguishable. The onset of light, the L1 and L2 cell, they hyperpolarize, and at the offset, they show this rebound depolarization. There's no distinction between those 2 cells. Now, for the off channel, an easy way of producing pathway specificity for off would be you simply rectify here, and you just take this rebound depolarization and transmit it to the next cell. So we took a look at that possibility by calcium imaging and expressing a genetically encoded indicator in the L2 cells. That was done by Dirk Reif, who is now in Freiburg, and we used the 2-photon microscope because otherwise your excitation light for the fluorophore would blind the fly. So you need to confine it along the C-axis very carefully. Where there's another problem with doing calcium imaging in the visual system of the fly, and that is that in order to detect these small signals from the terminals, you need to crank up your photomultiplier to be very sensitive. Now, if you are very sensitive and then you present a visual stimulus, these photons here kill your signal in the photomultiplier tube. Right? There's no way that you can present a high contrast stimulus and at the same time do sensitive measurements of fluorescence. So the idea that Dirk Reif came up with is simply present the stimulus only during flyback of the scan mirrors. That was very essential. Now, Dirk did these measurements from L2 terminals, and here's what he got. If you turn on the light, it's very little. If you turn it off, you get this huge off response. It looks very slow here. In fact, here you get the time bar. If you switch back to light level, then it's terminated rapidly, right? But this cell indeed shows a very high selectivity for the off edge of the luminance.
So what is the difference between this and the previous experiment, of the previous line?
Okay.
You showed.
This is electrophysiology recording from the dendrite of the L2 cell and L1 cell. So this is membrane potential. And now we're looking at calcium in the axon terminal of the cell.
So the input is not selective.
Exactly. The membrane potential of both cells, look identical, but the output is different.
What exactly is the off signal at the photoreceptor level?
Means that when I turn down the light.
But what exactly happens to the receptor molecule? I mean, what's the percentage
perception of off signal. The on signal is clear, light is absorbed.
Yeah.
Turn off the light. What exactly turn off the potential of signal?
Oh, okay. I mean,
there is dark current.
no.
I don't know.
No, there is no dark current in the fly. There is no dark current. No, simply you do not keep the channels open, so the membrane potential goes down to resting level in the photoreceptors again.
Passively.
Yes, yeah.
Fine, thank you.
Passively and rapidly.
Fine.
Yeah, okay. So that's the idea how the specificity is achieved for off in the L2 channel. So how about L1? So one idea, I mean we don't know yet, would be that L1 is inhibitory on a postsynaptic neuron. So in this hypothetical postsynaptic neuron, the signal would look like that. Now then, in this postsynaptic neuron, you simply do the same trick that we did with L2 and, rectify it, and then we're left with a positive signal at the light increment only. In fact, this is not completely hypothetical. There are some data that support that. In fact, I have seen recordings from a postsynaptic neuron to L1 from Claude Desblanc's lab in New York, and it looks like this. So, very much. And Tom Glendinning had done calcium measurements of L1 cells as well, and this is L1 response and to a light edge and a dark edge, and you see that L1, the calcium signal is fairly symmetrical, whereas in L2 cells, that's the blue, here you see that strong asymmetry with respect to the dark edge in L2. So that's, just one hypothesis how this is being done, but we have the functional data that at the end, L1 carries on, L2 carries the off signal.
But it's not but according to this, it's not the L1 and 2.
No, it's
one
downstream.
pathway, yeah.
Exactly. It's pathway. L1 and L2 sort of are the entry gates to the two pathways, and sort of the specificity is created either in the postsynaptic, in the axon terminal of L2, or in case of L1, postsynaptic to that. So next question is how many detectors do we have now per column? According to the sign rule of multiplication, if you split up the input signal into an on and an off, you would need 4 detectors per column to take care of all possible combinations, on, off on, and off. That would be the equivalent of a sign-correct multiplication identical to this Reichardt scheme with unrectified input. But maybe the fly sort of gets away with just 2 detectors, only on, and off. Now, in order to decide between that, we used apparent motion stimuli where we had light pulses in adjacent areas of the fly visual field separated by some time. And then the 2 models, the 4-quadrant and the 2-quadrant models, predict different things. The 4-quadrant detector predicts for on sequences positive, for off, positive, for on-off and off-on negative responses. These 2 are here. The 2-quadrant, sort of the reduced, only predict positive responses to on and off, and basically no responses to these mixed stimuli. Now, again, you see the people involved in that project here. We did recordings in blowflies and in Drosophila, and we saw both cases positive responses to on and off for Calipara, the blowfly, and Drosophila, and we saw no negative responses in response to these mixed stimuli, actually slightly positive responses in the blowfly. So from that, we conclude that we're dealing with only 2 detectors per column, 1 for each pathway. Now, there has been a competing model published in the paper back-to-back with ours from Tom Glendinning, and this is our sort of simple model, and that is the more complex model that this other study proposed. And so here you have on and off. but the L1 pathway carries this mixed off-on, and the L2 pathway carries these on-off detectors in addition. And from the previous experiment, you can already discard the existence of these mixed detectors because we did not see any responses to on-off and off-on. But in order to decide between these two models, we did some further experiments where we again presented apparent motion stimuli, but this time recorded in L1 and L2 blocked flies. Now, our model predicts, again, positive responses to on-on and off-off, as does this Clendenin model. But for L1 block, we would say that specifically the on-on sequence is reduced while the off-off sequence is unaffected. Now, the Clendenin model says both are reduced by the same amount. If we block L2, our model says on-on responses are unaffected and off-off responses are specifically reduced. The Clendenin model says, again, both responses are reduced by 50%. So we did the experiments. Here are the responses of the control flies and of the L1 and L2 block flies, and you see that you get nice positive responses to on and off. And now we do the L1 and L2 block, and when we block L1, the response to on is completely gone, while the response to off is still nice and healthy. And if we block L2, the opposite is true. We still get a nice on response, but no off response, right? So we're pretty sure that's the case. So last question is what neurons are postsynaptic to L1 and L2? In fact, that is sort of one of the most important consequences of this L1 and L2 finding, that we now can use anatomy to guide the next steps. Simply look which cells are postsynaptic to L1 and L2. These are then sort of prime candidates for the interesting computations that are taking place in the Reichardt detector. And a lot of anatomy has been known, and even more is being produced these days. Ian Meinertzhagen and Mitya Tzlowski at Geneva Farm do serial sectioning, TEM, provide nice connectivity data in the fly optic lobe, and tell us what neurons are really postsynaptic to them. And from their work, we know that postsynaptic to L1, there's one cell called TM3, and there's another one called MI1, and they seem to synapse on the T4 cells, which then, I said, bring the signal to the lobular plate. And the L2 pathway, there are also prime candidates called TM1 and TM2. They both terminate in the lobula, and then the signal is brought to the lobular plate by the T5 cell, right? So the obvious idea, from looking at the anatomy was that T4 cells represent the output of the on-motion pathway, and T5 cells the output of the off-motion pathway. And now, not with respect to on and off motion, but the idea that T4 and T5 cells bring the signal to the lobular plate is actually very old. And first of all, people have observed from Golgi studies that there are 4 subtypes of T4 and T5 cells which ramify in selective layers of the lobular plate. Now, these layers are obvious if you look at immunostaining by disc large. For example, you see 1, 2, 3, 4 different layers of the lobular plate. That's a horizontal section now. Right? And if you look at the dendrites of the cells, you find that HS cells ramify in layer 1. My pointer is dying. So here, and VS cells terminate in this most posterior layer. And from Erich Buchner's 2D labeling back in '84, we know that different directions of motion label different layers of the lobular plate. So that was all sort of circumstantial evidence pointing to T4 and T5 cells, and we thought we should now bring sort of stronger evidence, and we again relied on a nice driver line that we got from Gerald Rubin and Alyosha Nern from Geneva Farm, which labeled both the T4 and the T5 cells, and you see from that staining here how nicely they stratify here in the 4 different layers of the lobular plate. And this is the work of Bettina and Sham Raghu. He's in Singapore now. She's in with Michael Dickinson in Seattle. And when we now silence T4 and T5 cells, we again are able to abolish the motion response completely. That is the response of a HS cell, in fact, a VS cell of the control flies. You get preferred a null direction response, and when you then do the temperature shift in the T4 and T5 cells, then the motion response is completely gone. That is a quantification. These are the responses of 2 types of control flies, and these are the motion responses to preferred and null direction of the experimental flies where we blocked the synaptic output from T4 and T5 cells. So this shows that these T4 and T5 cells indeed provide the input to the tangential cells. Of course, one needs to know now whether indeed T4 specifically are the output of the on-motion detectors and T5 of the off-motion detectors. In order to test that, we need specific driver lines for either T4 and T5 cells. We're working on that. We don't know yet at the moment. So let me just sort of summarize where we are in our search of the Holy Grail. First discovery we made 2 years ago is that the actual Reichardt detector is duplicated in the fly visual system by splitting the input into an on and off pathway and then doing this calculation in parallel channels, one for the on, one for the off. Then we know that the L1 cells represent the input to the on pathway. Here you see the anatomy of the cell. The L2 cell represents the input to the off-motion pathway, and we have identified T4 and T5 cells as the output cells of these local motion detectors providing input to the tangential cells. Of course, we are not there yet, and the big question still is what neurons are in between L1 and L2, and what of them, which cell is doing what kind of computation, which is the delay line or temporal low-pass filter, where is the multiplication going on. So this will keep us busy for the next few years. We are going to proceed with, having more specific driver lines, like for MI1 and TM2. These are some promising driver lines selected for T4 and T5 cells that we're currently working on, and we use the same paradigm that I, showed to you over and over again, that we express Shibiri or KIR, the potassium inward rectifying channel, in these different cell types while we record the motion response with the patch pipette from the tangential cells. That is sort of easy. It's, doesn't need much brain at the moment. You just go through the different candidate neurons, you record here, and then you see whether neuron X is participating in the response or not. It's pretty clear cut. It's a lot of work, and we're doing it right now. Another approach that is more sophisticated is the use of channel rhodopsin for circuit analysis in the fly. Now we're expressing these channel rhodopsin not in the tangential cells, but in presynaptic neurons that we think are presynaptic to the tangential cells, while recording electrically from the tangential cells. And that is the work of Alex Maus in the lab. He is now stimulating T4 and T5 cells optogenetically with channel rhodopsin 2C128S, this bistable switch thing, and then he can elicit nice EPSPs and IPSPs in the tangential cells, and now he's combining that with pharmacology in order to work out what transmitter systems are involved there. And finally, we want to record from these various types of cells, just record the visual response property using genetically encoded indicators like GCAM3 or T and XXL. Examples for that I have shown to you with the recording from L2 cells. And that is our strategy for the next couple of years in order to uncover the cellular implementation of the Reichardt detector, that has been hidden in the jungle of the fly optic globe for more than 50 years. Thanks a lot.
So these flies that are mutated for the
Shibiri.
Shibiri, can they fly?
Depends on, this depends on how specific your driver line is. It might be that the driver line is very specific for the optic lobe. It only sort of targets, say, one cell type there. But in addition, it has strong expression in the thoracic ganglion or central brain somewhere. So the fly is paralyzed, cannot fly. That can be the case. It does not hurt our experiments while we do electrophysiology on the tangential cells because it's specific for the optic lobe, does not label any other cell there, but we cannot do behavior because it has additional expression, say, in the thoracic ganglion. But if you're lucky, it doesn't. It is really specific, throughout the nervous system. Then you can do behavioral experiments.
But you said that the Berkeley group did behavioral experiments.
So yes, those cells, those flies are healthy, right? Even if you express Shibiri, they still can walk and
But what is the effect of blocking L1 or L2
on the behavior?
The question is if they can fly, because
sure.
the whole idea of the optic flow is that they use it for flight.
Yes, or walking, whatever, right?
But they both on and off in the moving.
Yeah. So this is the Stanford group, Tom Clendinin. What they did was they used a styrofoam ball where the fly is walking on. So very often, sort of, even if the fly is not that healthy anymore, walking is still okay. Flying is forbidden. So they presented these on edges and off edges to the walking fly, and they showed specific reduction of the responses depending on whether L1 or L2 is blocked. Behavioral responses, exactly. But the results from these behavioral responses do not agree exactly with our electrophysiological recordings. And from this discrepancy, they sort of came up with the more complex model to account for it. And for those of you who want to know more, I have sort of more slides. It's getting a little bit sort of complicated and sophisticated, but I can explain to you what the exact difference between their model and ours is.
But just a follow-up. But you simplify model. Can you make it consistent with the behavior? Only so you have to assume that the blocking is affecting other parts of the system.
Yeah, I mean.
Or right now it's a question mark.
It's a question mark, I would say. Just in general, I mean. In general, if you look at behavior, there are other systems at work simultaneously. They will not, it's not the case that the fly has only the optomotor cause control system. The fly needs to find objects and everything, and so, we expect that there are other systems in parallel to the optomotor system, and so all that will show up in behavior, right? For example, I mentioned that to Jeff Kemhai 2 days ago. If we look at the behavior of these T4, T5 block cell flies, right, their response to large field motion is 0. They don't show any optomotor response. It's completely blocked. If you give them, in a feedback closed loop experiment, control over a single bar so they can keep it anywhere they want, so to say, then they do a very nice fixation response, like the wild type. So they're not blind, but their large field course control system is dead, right? So there's more, in behavior you get all of that.
Thank you very much. Yeah, sorry.
Okay.